Okay, people can join while I introduce everything. So, hello everyone. Welcome to the third seminar in our series on integrating speciation research. And this is part of a kind of output of a European Society for Evolutionary Biology funded special topic network, with the aim of kind of integrating speciation research. And one of the goals of this network is to put on a kind of seminar series as we're doing now. And so this is the final talk on our first topic, focused on bridging macroevolutionary patterns and microevolutionary processes. And today we're going to take a comparative perspective on this problem with talks from Hélène Morlon and Dan Rabosky. And so just a few quick reminders before we start. If you do have questions, feel free to please post these questions in the chat and we'll put those to our speakers. And so this first 1 hour will be both talk sessions from the speakers. And after this first 1 hour session, it will be followed by a discussion session, which will stay in this same link for, which will be a more general discussion on how we might bridge these micro to macro evolutionary scales. And so with that, it's my pleasure to introduce today's first speaker, Dr. Helen Mullon. Helen is a CNRS Research Director and TED Fellow at the Institute of Biology at the École Normale Supérieure in Paris. Her research combines mathematics, bioinformatics, and fieldwork to study a range of questions spanning macroevolution and macroecology, and also through to community assembly, biogeography, and conservation. And so in particular, Hélène works at the cutting edge of comparative methods development and regularly develops new phylogenetic models to test hypotheses on the tempo and modes of species diversification. We're delighted to have her with us here today, and she's going to talk to us about towards process-based comparative models for bridging micro and macro evolutionary speciation research. Thanks very much, Hélène. Please take it away.
Great. Thank you very much for organizing this series of seminars and having me here, and thanks for the introduction. So my own interest in speciation research comes from understanding patterns of species richness. So why some groups of organisms are more species rich than others, when some regions of the planet are more species rich than others. So that's where my interest in speciation research comes from, and why it's super important is because we can more or less explain with a macro evolutionary perspective present-day levels of species richness in species groups or particular regions as the balance between speciation and extinction events. So when we have this perspective, then there are two particular biological parameters that are super important, which are the rate of speciation and the rate of extinction. So because those are the parameters that, to some extent, explain present-day species richness, what you're going to really clarify what those are. So the speciation rate is the average number of speciation events that can occur per million year per lineage. And so really simply, if we have groups that have the same age and some with higher speciation rates than others, then we would expect on average that they would lead to higher species richness today. One of the ways that people had to approach this question is to use comparative methods. As most of you here are really familiar with, we can come up with estimates of speciation and extinction rates and how they might vary through time across lineages and so on by fitting stochastic birth-death models to reconstructed phylogenies. And there's a lot of developments that I've done along these lines. I have references here that are reviews, but of course, Dan will present after, also had made a lot of developments in this field. But we have a battery of models that allow us to fit those types of models to reconstructed phylogenies and get at estimates of speciation or extinction rates. And what We have seen when fitting those types of models, and in particular models that allow for rates to vary across lineages, is that there is a wide variation in speciation rates across lineages. So if we look at this particular clade of birds here that I have on the left, the speciation rate can vary by 2 orders of magnitude. So from 0.01 event per million year per lineage to almost 1 event per million year per lineage. So a wide variation. And when we look at the scale of all birds, there can be as much as 3 orders of magnitude variation in this rate of speciation. What has been an area of active development in the field is to try to understand what are the factors that modulate speciation rates. And in particular, people have worked on 2 axes, either looking at particular interesting features of a species, so species-specific traits like reproduction mode, life history traits, dispersal capacity, and so on, that are all characteristic of a species that can modulate the speciation rates. And they've also looked at the interaction between these species-specific traits and extracting factors such as variation in the climate or the geological context, or also biotic factors such as the effect of interspecific competition, mutualistic and antagonistic interactions, and so on, that can all interplay to modulate rates of speciation across lineages. Now there's always like several levels of understanding of what might, of the processes that might control speciation rates. And if we really want to understand how abiotic and biotic factors in combination with species-specific traits really influence speciation rates, then we have to understand how they influence the speciation process. And that's where we can start to look under different modes of speciation, how speciation occurs from an original population to an initial step of the speciation process where subpopulations start to be differentiated, to the evolution of reproductive isolation, and finally speciation. And there's been a particular focus on at least on macroevolutionary studies of speciation, on how the evolution of reproductive isolation occurs, at which rate, and so on. And what then has been done is to try to start to see what is the correlation, what is the association between this evolution or measures of the evolution of reproductive isolation and speciation rates measured at the macroevolutionary scale with the type of models that I talked about. Dan is going probably to present a lot of this research. So just a quick slide to say that these results have, of this analysis have allowed to document how measures at the intraspecific level of genetic differentiation or reproductive variation and so on actually correlate with speciation rates estimated at the macroevolutionary scale. And there's mixed support with sometimes results of decoupling between some of these measures and speciation rates, sometimes positive association, and so on. So we carried one analysis in this direction in my research group, where we tried to document and to test the association or the correlation between intraspecific genetic diversity and speciation rates across mammals. So this is a research that has been carried by Anna Silva, mostly in collaboration with other people. And so why genetic diversity? So we decided to look at this association between genetic diversity and speciation rate because genetic diversity starts to, or at least measures of genetic diversity starts to be available at the level of whole clades. So we can actually do these types of start to do these types of analysis and document these patterns. And then genetic diversity is also supposed to correlate with a bunch of metrics that we are interested in. So things like the supply of genetic variation that we have in populations, reproductive isolation, geographic structure, all processes that are supposed to positively impact rates of speciation. So the expectation we might have is that under geographic or ecological models of speciation, we might expect to see a positive association between genetic diversity and speciation rate. This association hasn't been tested a lot in the literature. We found one study of this association in fungi that find a negative and weak correlation between effective population size or genetic diversity and speciation rates. So Anna put together a pipeline to assemble as big of a data set possible to compute genetic diversity across mammalian species. And so she ended up having measures of genetic diversity for more than 2,000 mammalian species with at least 5 individuals. So now we can run the type of models that I presented, where we can have estimates of lineage, specific estimates of speciation rates. So this is done here with our own implementation of our own development of a model that allows to estimate speciation rates at the level of lineages. The CLADES model shown on the phylogeny, and then on the right we have the estimates of genetic diversity that are obtained with either Watterson's data or an ASPI measure. And we see that what we found is regardless of the different measures of genetic diversity that we used and a bunch of robustness analysis that Anna carried, whatever we did is we found support for a negative association between genetic diversity and speciation rate, and this at the level of all mammals, and also when we look at particular mammalian groups, also a negative association for almost all of them, more or less significant, but strong tendency for this negative relationship. So then we try to understand where this negative association might come from, given from the beginning we were more expecting to find a positive correlation. So we thought that this might be linked to specific characteristics of the species, where we would have positive or negative association between genetic diversity and the specific and these characteristics of the species, and the other relationship between speciation rate and these characteristics that would lead indirectly to a negative relationship between genetic diversity and speciation rate. So we found that these particular traits, so for example, body mass, range area, and so on, some of them did correlate with genetic diversity, but we didn't find a significant association with speciation rates and these particular traits. And also, when we looked at the association between genetic diversity and speciation rate while accounting for variation in trait values, we didn't find, we found that the relationship hold. So it doesn't seem like at least with the traits that we measured, that they did not explain this negative association between genetic diversity and speciation rate. So then the other hypothesis we might have is that it's not actually like the supply of genetic variation and genetic structure and genetic diversity that's going to foster fast speciation, but it's more the process of speciation that acts on by demographic effects on genetic diversity. So at speciation, there is a reduction of population size that might limit species genetic diversity, and we can maybe think that if speciation is fast enough, there won't be enough time for genetic diversity to build up again, and so it would be speciation that exerts the limits on species genetic diversity rather than the other way around. So this idea that rapid speciation might limit the accumulation of genetic diversity. The other hypothesis could be that that geographic structure and reproductive isolation actually does positively correlate with speciation, but that genetic diversity is inversely related to geographic structure and reproductive isolation rather than the other way around. And it has actually been proposed under some models that are different from the continent island model, that we can indeed have an inverse relationship between genetic diversity and geographic structure or reproductive isolation. So that's also a possibility. And Finally, another hypothesis that we could think about, maybe there are others, is that we could actually expect to see more speciation when in species that have a small rather than large effective population size, with the effect of reproductive incompatibilities that might accumulate faster under a scenario of smaller population size with a reduced efficiency of purifying selection. So this has been proposed also recently, that this model, this geographic model of speciation might be more important than we think. And under this hypothesis, we would expect to see a negative relationship between effective population size and speciation rate, which in the paper has been tested through measures of range size rather than genetic diversity, but it could be this effect that we see here. So to conclude on this research or this on this project, what we found was a pretty robust and consistent negative relationship between genetic diversity and speciation rate. But then we're left a little bit with this pattern, and it's hard to distinguish the different. We did some tests to see which of these hypotheses might be the most supported, but there's still like a bunch of uncertainty on whether we have something like speciation exerting a limit on species genetic diversity, or genetic diversity being inversely related to population isolation, or this demographic effect of speciation, where species with small rather than large effective population sizes would accumulate reproductive incompatibilities faster. So I think this example reflects the difficulties we have in testing which microevolutionary processes might act as a rate-limiting step in speciation when we use correlative approaches. And so one of these difficulties is that it's hard to know at which stage of the speciation cycle we measure genetic diversity. So when we measure genetic diversity or genetic differentiation or population isolation across species in large clades, we might have, at present, we might have some species that are in the initial step of speciation, where we might expect to see high genetic diversity because we have a lot of polymorphism and differentiation of populations, or we might measure genetic diversity when speciation just occurred in a situation where we have low genetic diversity in newly formed species, in particular with this effect of this demographic effect of reduction in population sizes. So this reciprocal effect of speciation on the macroevolutionary measures of differentiation that we use complicates this interpretation of correlations that we might see at present. Also, when we measure speciation rate using comparative models on phylogenies, we also can wonder what are we actually measuring when we do that. And in these models, what happens is that we don't model the speciation process. And so speciation is just considered to be an instantaneous event where we have two populations of the same ancestral species that give rise to two distinct descendant species. So it's an instantaneous event that just happened with a rate, and so it doesn't really correspond to this idea of speciation, where we need to have first a speciation initiation, then the evolution of reproductive isolation, and also the survival of incipient species until speciation completion. So there's when we see the wide variation of speciation rates that we observe across phylogenetic trees, we might wonder which of these three major aspects of speciation drive variation in speciation rates. Is it speciation initiation that's super variable across species? Is it the evolution of reproductive isolation? Is it the survival of incipient species? All these three aspects can act as a rate limiting step at the macroevolutionary level, but we only look at it as a single instantaneous event. So there's one macroevolutionary model that has been developed that accounts a little bit more for these three aspects of the speciation process. So this model is known as the protracted speciation model. And so I just thought about how much this model could help us to bridge micro and macroevolutionary speciation research by distinguishing these three aspects of the speciation process. So in this model, what we have either true species or incipient species, and there is a rate at which speciation initiates, a rate at which speciation complete, and then also extinction rates. So when we start speciation, we first have incipient species. So we have speciation initiation here. During this time period, we have evolution of reproductive isolation, but no true species. And finally, we have speciation completion when we really have two true species that have evolved reproductive isolation. So now when we see what we can actually estimate by fitting these models to comparative data, what we see is that we can estimate the duration of speciation, but we cannot estimate the speciation initiation and extinction rates. So in terms of then having a better understanding of a different or better estimation of the different stages of speciation, it's not that straightforward, but things that haven't been explored yet are, for example, how much or whether, for example, adding intraspecific genetic data could help us in estimating these different rates that then would help us to better understand which aspects limit speciation at the macroevolutionary scale. And also this model hasn't been developed in the way that other models have been developed, where we can have, for example, species-specific estimates of the rates. So there's a bunch of developments that would be required also to be able to have the same types of measurement of the macroevolutionary scale of these different stages of speciation. And even though it's a model that has some advantages in comparison with other models where we just have an instantaneous rates of speciation, it does remain phenomenological. So we don't have a modeling of demography and population genetics and intraspecific genetic differentiation processes that might occur when we have the evolution of reproductive isolation. So something we're starting to do is to try to build these macroevolutionary models that would account more for the interplay between speciation and both demography and intraspecific genetic differentiation. At the moment, what we have is more something where there is not yet a feedback between the demographic processes and population genetics to the speciation process. And we first built this model to try to see whether our hypothesis that speciation could in itself, the effect of speciation on demography could in itself explain the negative relationship that we observed between genetic diversity and speciation rate. So here what we have is at the macroevolutionary level, a process such as the one we used in macroevolution. So we have a lineage-based model. So it's the class model here that we use, where we have an initial speciation rate, and then when speciation occurs, there is a modification of the rate of daughter species, and these rates are drawn from a log normal distribution. So this allows to, at the same time, generate some heritability of the rates, but also some heterogeneity, so stochasticity and variation in lineage-specific rates. And on these macroevolutionary processes, what we do is that we simulate demographic processes. So here we took a model where each species follows a density-dependent population dynamics, where growth rates evolve as a Brownian process. We have naturally a random split of individuals as speciation, and then extinction naturally occurs when we have the death of all individuals in a given species. So once we have demography added to this macroevolutionary model, we can infer levels of genetic diversity by using coalescent models or other types of models in population genetics that allow to have expectations for genetic diversity at the species level. Okay, so as I said here, we don't have a feedback between, so we have a speciation process that is going to influence demography and population genetics, but we don't have a feedback of these processes on the speciation process. And what we found is that this model can either predict a positive or a negative association between speciation rate and genetic diversity, depending on the relative pace of speciation and the accumulation of genetic diversity. So it's reproduces this hypothesis that we had in mind, where if speciation occurs rapidly, then it can have somehow a control on genetic diversity via demography. There's a whole range of parameters for which, and a wider range of parameters for which we can, with which we obtain this negative correlation between genetic diversity and speciation rate than parameters for which we predict a positive relationship. And this model can be fitted to data, in this case, not using the classical maximum likelihood or Bayesian inference that we usually use, but using machine learning techniques. So, I think that, like, really, if or something that would really allow us to maybe have a better understanding of these types of correlative patterns that we observe when we correlate intraspecific measures of population differentiation and speciation rate, would be a model where we have these three processes of speciation initiation, evolution of reproductive isolation, and speciation completion running and influencing demographic processes and population genetics, such as what I have shown in the other model, but also where these processes can retroact on the rates of speciation initiation, and in particular evolution of reproductive isolation and speciation completion. So yeah, I think those are my conclusions. We start to be able to document these patterns, and it's really great, and we can find unexpected results or things that we were not, that can be surprising. And there's a bunch of developments on the side of developing comparative models that can still be done to better understand which aspect of microevolution act as a rate-limited step in speciation, and finally how what explains patterns of species richness that we observe today. With that, I would want to thank you for listening, and the people we have been involved in this work, as well as funding sources.
Thank you very much, Helen. That was brilliant. So, yeah, please, if anybody has any questions, feel free to post them in the chat. So we have one actually that's just come in from Louis, who asks, for fitting multi-predictor models of speciation rate, does PGLS have limitations? Can better approaches be developed? I'm not sure what that QAA power and false discovery rates. So in the results that I've shown, we use PGLS, and then we use the Bayesian approach, PBLM. Yeah, those certainly have limitations. I don't have a lot of things to say along these lines. The statistical approach is to relate speciation rates to other predictors while accounting for multiple predictors. I don't know if there's better. Yeah, I don't know if specific limitations have been shown. Great. So one from Dan: are the data rich enough to distinguish non-equilibrium demography? By which I mean, can low genetic diversity be synonymous with has undergone recent range expansion?
So I mean, it's are we talking about the mammalian genetic diversity, or in general, do we have enough? So I think there's in phylogeography, there's data that's available that's not only about just having an alignment of 5 individuals, as we did here, where we measure genetic diversity and where we can distinguish different things like either demography or other processes that are going to explain genetic diversity. So there's a lot of models in population genetics that allow to do much better. Now, when in our case, we just have this measurement of genetic diversity from an alignment of 5 individuals, yes, it's a big statement to say that low genetic diversity could be synonymous with recent range expansion. It's just like one potential explanation or one potential interpretation of differences in genetic diversity and across species and how they might correlate with speciation rate, but under a bunch of hypotheses, yes, of course.
Great. Probably just time for one really quick one just before we switch over. So Anya asked, for including demography, how do you include parameters like carrying capacity?
Okay, where is this one?
Third one down.
So do you have a correlation between speciation and genetic diversity? What was the definition of a species? No, not this one, sorry. Genetic diversity, I don't know if
so from Anya,
for including demography, or do you include parameters like carrying capacity? Carrying capacity in this particular model as its state of development, it's fixed. So we evolve growth rate, but we don't evolve carrying capacities. But I mean, it's a free parameter, right? So it can be varied in the models, it can be estimated, but it's not, it doesn't vary in the current development of the model.
Great, thanks very much, Helen. Thanks for all those questions. So I'll pass over to Liz, who's going to introduce Dan. Thanks very much.
Great. Thanks so much, everyone. And we'll try to get back to some of these other questions in the discussion, but let's stay on track here with Dan's talk. And so Dan, you can go ahead and share your screen when you're ready. So it's my great pleasure to introduce our second speaker today, Dr. Dan Rabosky. Dr. Rabosky is an associate professor at the University of Michigan, as well as an associate curator at the University of Michigan Museum of Zoology. Dr. Rabosky's research seeks to understand the causes of variation in rates of diversification and trait evolution across the tree of life, as well as the effects of diversification rate variation on the structure of ecological communities and spatial patterns of species richness. His research fuses cutting-edge mathematical theory and statistical model development with the deep knowledge of natural history and biogeography, particularly of squamate reptiles. His talk today is entitled Microevolution, Macroevolution, and the Origin of Species. Can we make speciation a more predictive scientific theory? So go ahead whenever you're ready, Dan. You're still muted.
Okay, yeah. Now, thanks, Liz and Chris and Joanna and everyone else involved in the organizing this special topics network, and also thanks to the European Society for Evolutionary Biology for organizing it, and also to all the other speakers for an overall a very stimulating set of talks. So what I'm going to talk about today is whether we can make speciation a more predictive theory. And so this isn't really a talk that is rooted in data or new results, but rather a talk that is more of a personal perspective on and speculation about some topics in speciation. And I just want to start with a couple of quotes about scientific theories more generally and the role of prediction. And this is a quote from the first one's a quote from the U.S. National Academy of Sciences. One of the most useful properties of scientific theories is that they can be used to make predictions about natural events or phenomena that have not yet been observed. And then one from Haldane, which says that in scientific thought, we adopt the simplest theory, which will explain all the facts under consideration and enable us to predict new facts of the same kind. And so what I want to ask today is whether the theory of speciation is a predictive scientific theory. And by theory in this case, I mean theory in a broad sense, not mathematical theory per se, but rather the general explanatory principles and predictions that emerge from our collective understanding of the speciation process. And another way of viewing this is thinking about Is our knowledge of speciation give us the ability to take some attribute that we know of species or populations or genomes and predict some property of speciation? And I think this level of predictive accuracy is really necessary before we can say claim that speciation has the same sort of explanatory power that some of the other major tenets of Darwinian evolution would have, such as the explanatory power of common ancestry or natural selection. And so back to this main question of whether speciation is a predictive scientific theory, I'll go through some reasons, evaluate some aspects of it, the idea of speciation, which it may or may not be predictive, and then I'll speculate at the end about some ways in which we might be able to make the theory a little bit more predictive. Now, before starting, I want to be a little clear about what I mean by the idea of speciation rate. I think this is a question that is at the differences of interpreting this idea are at the heart of some of the potential disconnects between microevolution and macroevolution. And I think that actually the idea of what is speciation rate is in some ways a more general, it can be framed more generally as differences in how we might even think about speciation at different scales, microevolutionary and macroevolutionary scales. And just to give you some idea of what I mean by that sort of either disconnect or the differences that we see across scales, I want to turn to a well-studied example, a model system in speciation research, three-spine stickleback. And I'm picking this example not because I'm picking on stickleback, but rather because I think this is a very well-studied system that illustrates many of the challenges that we face as biologists in conceptualizing speciation and speciation rate across scales. So moving from the population scale to the sort of macroevolutionary and phylogenetic scale. And I would argue that the patterns and challenges that we see in linking those scales across stickleback apply much more generally across the tree of life. And so when we think about stickleback, it's worth keeping in mind that even though stickleback are capable of evolving reproductive isolation very quickly in some contexts, in the broad sense diversity of stickleback, there are about 20 species of taxonomically distinct entities in that family. And so if you want to think about those for now as good species, which I'll get to in a second. And so if we looked at this diversity in a phylogenetic framework, it takes about 5 to 12 million years for each new taxonomic species to arise. So this is sort of a phylogeny of the Gasterosteidae. But if we were to look within a single entity, such as the three-spine stickleback, we might find that we could elevate many, many more populations within that taxon as to the species level under the biological species concepts. They have attributes that would mean that we could, in principle, look at some of these diverging populations and elevate them. And if we were to ask what our perspective on biodiversity might look like, were we to do that, we might see something that looks much more like this, where we have this smattering of recently diverged populations at the tips of the tree. And so I like to think about this as sort of when we think about these populations at the tips of the tree, of course, many or most of these new species or these populations are not going to persist through geological time. And at the same time, it's these deeper splits in the tree, or these are these speciation events that might be contributing more to biological diversity overall. Now, I think I like to think about this problem as a good species, incipient species continuum problem. And we often don't know where we sit on this sort of scale. I think that the way in which we talk about biodiversity at microevolutionary and macroevolutionary scales is contingent on taking some particular, on whether we are working at the sort of good species level or the incipient species level. And this is, of course, not a new problem in biology. This is something that goes way back to Darwin, who writes in The Origin, "It need not be supposed that all varieties or incipient species necessarily attain the rank of species." And Darwin says, well, they may, whilst in this incipient state, become extinct, or they may endure as varieties for very long periods. And so, of course, at some point they go on to become what Darwin would also call tolerably well-defined objects. And so all of this is a little bit of context to this idea of what I mean by speciation rate. So what is speciation rate? So this is a time-calibrated phylogeny of what we might think of as good species. So these are our New World wood warblers in the family Parulidae. And so when I talk about speciation rate, for example, I'm typically meaning something like, based on our past history, what is our best estimate of the rate at which so-called good species give rise to more good species, whatever those good species are. And we can take a tree like this, we can, as Hélène pointed out in her talk, we can fit evolutionary models of speciation and extinction to these types of data and obtain estimates of speciation rates. For example, in this case, we might estimate rates of speciation rates at the tips of the phylogenetic tree. Here I'm showing you potential speciation rates estimates for a set of individual species at the tips of this tree. And this is really the rate at which good species produce more good species, which is quite an unsatisfying definition in some ways, because it raises questions that we often don't wrestle with directly, which are, what are these good species and how do they relate to incipient or ungood species? I don't have any answers. All I can offer is to contextualize that observation and more or less raise the fact that this is an issue that we have to continue wrestling with. And it's not necessarily something that even has a clear answer, but it is something we should be aware of when we talk about speciation at different scales. So, is speciation a predictive theory, and can we make it more predictive? All right, so I think there are three general areas that I'm going to outline where I think we have and continue to make predictions using what I think of as the general explanatory body of theory that is speciation. And the first is that speciation theory makes explicit predictions about the amounts of reproductive isolation or the rate at which reproductive isolation accumulates. So for example, we can go back to Coyne and Orr's classic studies on the tempo of accumulation of reproductive isolation as a function of evolutionary distinctiveness of taxa. And so we have a lot of reasons to think now that hybrid incompatibilities and even pre-mating isolation will accumulate as a function of evolutionary time. So this is a predictive aspect of modern speciation theory. And there are also extensive amounts of evidence. There's also extensive evidence that reproductive isolation is frequently coupled to ecological divergence. So we have good reasons to think, and a lot of empirical evidence for a coupling between different components of reproductive isolation and ecological divergence in focal taxa. So this is a table from Cervetti et al. 2011, sort of summarizing some of these cases where there are traits that are associated both with reproductive isolation or assortative mating and ecological divergence. So there's a lot of literature on this. I'm not going to summarize it, but I do want to say, with respect to this predictive aspect of speciation, that if speciation is something more than the evolution of reproductive isolation, then those predictions might have limited relevance for the origins of biological diversity more generally. So if speciation involves things like the formation of isolated populations and the persistence of isolated populations, if those are critical ingredients of the speciation process, this is sort of an expanded scope of speciation that goes beyond reproductive isolation per se. And it means that these features that we can predict in terms of reproductive isolation might not be the key ingredients of speciation. And of course, this is not an original idea. This is an idea that goes back to Meyer and even earlier. Meyer talks explicitly about this in Animal Species and Evolution, about these other sorts of demographic factors that might affect speciation beyond just what his focus on isolating mechanisms. So another major area where we might say that there are predictions from the modern body of speciation theory and a pattern that we see with respect to speciation in nature might be the ways in which our speciation theory makes predictions about patterns of trait divergence or geographic overlap between closely related species pairs. And a lot of this emerges from, you can think about the geographic overlap predictions, for example, as emerging very simply from the logic that underlies the accumulation of Dubchansky and Mueller type incompatibilities, which does make predictions about how reduced fitness in a heterozygote state should relate to the difficulties of splitting populations in sympatry. And so Ernst Mayr writes clearly about that. And of course, we have seen that for many taxa, there's extensive evidence for allopatry of recently diverged sister species. So this would seem at first glance to be like a clear success of a very simple principle of genetic organization with respect to the structure of reproductive isolating factors and what it might predict about the geographic mode of speciation. And so this is a figure illustrating the frequency of sister pairs of birds, for example, that are sympatric versus allopatric. And you can see that most of these sister pairs of birds fall into the allopatric category. But alas, there are contrasting patterns of geographic overlap in many other taxa. This is a result for Heliconius, for example. You can see that more recently diverged sister pairs are actually sympatric. And so I would, in some ways, maybe our perception of extensive allopatry is more a reflecting our bias towards certain vertebrate groups, for instance. And of course, there are even some groups of vertebrates, such as hemulid fishes, show a similar relatively high frequency of sympatry among closely related taxa, at least compared to birds. And so I guess the point here is that our generalizations often prove to be far less general than we would like. And with respect to trait differences, we might say, well, does something about speciation theory predict trait differences between closely related species? We do know, for instance, that in a lot of cases, sympatric species pairs differ more in ecologically relevant traits relative to sympatric species, or relative to allopatric species. But of course, these differences might not be causally related to speciation, so they might reflect post-speciation character displacement or other sorting processes. And consistent with what I told you about allopatry, we find that these generalizations also break down when we look at other sorts of taxa. So, for example, here's another example recently published looking at premating reproductive isolation in birds, arguing that finding that song divergence in some groups is sometimes lower in sympatric species pairs relative to allopatric species pairs. And so the take-home message here is that when we look at patterns of trait divergence in speciation theory, we see few consistent signals emerging across diverse groups of organisms. So this brings me to my third major category of predictions within speciation. And I think this can be subdivided into two major categories, first of all, whether we can predict whether speciation happens, and secondly, whether we can predict how much speciation happens. Now, for the first question, does speciation happen? There have been some nice predictive studies in this regard. So here's one from Kiesel-Barraclough, 2010, looking at how the capacity for gene flow as a species-specific or clade-specific trait predicts whether taxa, the area of islands on which taxa will undergo speciation. The idea being that taxa that have greater capacity for long-distance gene flow, such as bats or large mammals or flying butterflies, might require larger areas to become geographically isolated and thus undergo speciation. So overall, there does seem to be this predictive relationship here where the greater your genetic subdivision you have, such as something like a snail, is more likely to undergo speciation on a smaller island because it presumably can be isolated by relatively finer landscape features. And of course, there are other examples that we could point to. Here's a paper from Katie Wagner and Ole Seehausen and Luke Harmon looking at how a number of other types of traits, some extrinsic ecological traits and traits associated with sexual selection, predict speciation in cichlids. And they found that there is some effect of sexual selection proxies on the speciation probability, as Ole pointed out in his talk a couple of weeks ago. Now, however, I would say that overall we have had limited success in this regard. And another point here is that these require replicate systems where we can clearly determine when speciation does or doesn't happen, such as insular biotas, lakes, and islands. And actually, Ole gave a really good summary of some of the limits to this type of extrapolation, even within the cichlid system in his talk. So we have really good evidence that even when there's statistical predictability in this sense, there are many other radiations where you might look at and say, well, this clade should have undergone speciation within the lake, but it doesn't necessarily happen. And so this brings me to my final sort of line of evidence here, or a test where we might evaluate the predictive utility of speciation theory, and that's to ask, can we predict how much speciation happens? Now, by how much speciation happens, what better measure of how much speciation happens do we have than the speciation rate itself? So I view speciation rate as a nice measure of how much speciation is happening. And as Hélène pointed out in her talk, we frequently find within groups of organisms such as birds, approximately at least at a large phylogenetic scale, such as all birds or all fishes or all mammals, we find something on the scale of several orders of magnitude of speciation rate variation. So this is one of a paper from Helen's group on speciation rate variation across birds. And a number of other studies have found very similar sorts of results overall in terms of the overall spread and range, a magnitude of speciation rate variation across major groups. So these are some results from birds, mammals, fish, squamates, and amphibians from data from Cooney and Thomas a couple years ago. So we have extensive speciation rate variation across the tree of life. And we might ask, can we predict this variation with some of the features that we think should be predictive of speciation rate variation? Now, alas, what we tend to find is that the key microevolutionary processes that should predict speciation rate do so inconsistently or not at all. So this is a recent paper by Ben Freeman et al. Looking at how rates of premating isolation, meaning song discrimination in birds in this case, predict speciation rate. And what they find is that there's no effect of this measure of premating isolation on speciation rate in birds. So that's obviously a trait that one would expect a priori to have a strong predictive utility, speciation rate in birds. And likewise, in a number of other systems, this is an example from that we published a few years ago, looking at trying to measure rates of premating and postmating isolation in Drosophila and rates of postzygotic isolation in rates of evolution of postzygotic isolation in birds, looking at how that correlates with speciation rate. Again, we find very limited evidence. We find no evidence that these traits have any predictive utility in that sense for predicting broader patterns of speciation rate. And more recently, we've been looking at how propensities for population isolation might correlate with speciation rate. And we've looked at this in neotropical lizards and snakes, and we found no effect of population isolation proxies measured using genetic variables and speciation rate in lizards and snakes. We've looked at this in some Australian lizards as well. And again, no relationship between population isolation as measured by genetic population structure and speciation rate. So overall, again, we have this lack of signal of microevolutionary processes in predicting speciation rate. So is speciation a predictive theory? I think I'd like to submit the following general tests when we think about whether speciation rate is a predictive theory. So I call this the predictive speciation challenge. And I'd like to take a set of taxa, such as these two birds, and give them to, I'd submit that it's very difficult if you submitted these images of birds but stripped of all identifying taxonomic or phylogenetic information. So you're not allowed to know what clade these birds belong to. And if you have perfect knowledge of their ecology, their life history, their geography, and their complete genomes, but no information about taxonomy and phylogeny, could you predict which of these lineages is actually belonging to the clade that is undergoing faster rates of speciation? And so these two species of birds are small, insectivorous, warbler-like things that breed in dry pine forests in the western or southwestern United States. And just based on ecology and life history, you would be hard-pressed to predict that, in fact, the one on the right is a black-throated gray warbler, which belongs to a rapidly speciating clade of New World birds, and the one on the left is an olive warbler from a very different clade of birds, which is barely speciated at all. And you could replay this sort of game over and over and pick other examples of ecologically matched representative taxa. Well, the taxon on the left here is a cryopelagic small fish that lives in the Antarctic that feeds on krill and copepods, and the one on the right is a cryopelagic fish that also feeds on krill and copepods. And based on their basic ecology and life history, you would also be hard-pressed to predict that, in fact, the one on the left is a nototheniod, which is a member of a very fast speciating clade of fishes, and the one on the right is a gatid or a cod, which is a slow speciating clade of fishes. And you could play this game over and over again. One on the left here is a relatively flattened ant specialized lizard that lives in Australia, spiny. The one on the right is an ant specialized flattened lizard that is also spiny that lives in North America. And in this case, again, you would be largely unable to predict, even with perfect knowledge of their biology. I think the fact that the one on the left is a much slower speciating lineage relative to the one on the right. So I think the verdict here is that for me is that at this sort of scale, I think the theory of speciation that we have at the moment is not very predictive. So the last couple of minutes, I'm just going to summarize some of my views on how I think we can go about making speciation a little bit more predictive. So first of all, I think we need to expand our conceptualization of speciation. I think we need to move beyond the paradigm of speciation as reproductive isolation to one that includes population divergence, reproductive isolation, and especially the persistence of recently diverged populations. And different types of traits might act differently at facilitating different components of that process overall. And reproductive isolation is, of course, only one component of the speciation process in this sense. And maybe it's not even the key or rate limiting component. And this expanded view of speciation, I think, raises conceptual problems that we can't ignore. And just to be clear, this is not a new idea. This is something that goes back to Meyer, and perhaps even you can read elements of this even in Darwin's writings about speciation. But what do I mean by conceptual problems? Well, this is my second point, which is that if we take this more pluralistic view of speciation, especially including population persistence or the non-extinction of incipient species, we really can't ignore this good species, incipient species problem. So we have these things that we see in phylogenies at different scales. We have these deeper divergences and we have these shallow divergences. And we don't know whether these things that we are calling incipient species are even qualitatively the same as the things that we see that represent these persisting speciation events over deeper time. I don't have an answer here. All I can do is point this problem out. And third, I think that we need a clear framework for evaluating the predictive utility of alternative population level microevolutionary mechanisms. And in my view, speciation rate does provide the strongest and most general independent variable, this measure of how much speciation has happened. But it raises fundamental questions about the units, questions that we can't simply make go away, that have to do with whether things are good species or incipient species or persisting or non-persisting species. I don't know how to resolve this question, but I think it's at the heart of the disconnect between microevolutionary and macroevolutionary views of speciation. Now, I also think that we have to acknowledge that despite my view that speciation rate is going to be an important dependent variable, a response variable for testing the predictive utility of some of our theory in speciation, we have real limits to knowledge with macroevolutionary frameworks. And I think nowhere is this clearer than our attempts to address the problem of why some incipient species persist where others do not persist. By which I mean that this particular problem, which is one of the hardest ones, I believe, in evolutionary biology, the information that we need to solve that problem is information that falls right in the gap between observations that we have at historical and ecological timescales and those that we have at much broader macroevolutionary timescales, sort of phylogenetic or paleontological timescales. So this is a hard problem simply by virtue of the timescale over which the processes play out. Paleontological information is often difficult to use because it's often too coarse or too time averaged or too spatially restricted. And so we have minimal direct information overall about the fate of non-persisting lineages, which obviously makes it difficult to understand why some incipient things persist when others don't persist. And I also think that we have a problem with our phylogenetic perspectives in that we're increasingly finding that phylogenies are only weakly informative about process. And so I think there's an increasing amount of evidence that we may be limited in the types of major insights that we can derive from about lower level processes from fitting better models to phylogenetic trees. And so this is an example of a paper by Jing-Chun Lee et al. From a few years ago that using simulation showed that different combinations of population splitting and persistence parameters could essentially give you very similar phylogenetic patterns, which isn't all that surprising given the information content of that data type. So my final thought here is that maybe we need a big science approach to synthesis in speciation. So I think that we have to recognize that persistence is a fundamentally hard problem, but we can generate large comparative data sets on reproductive isolation and population structure across diverse taxa. We already have information on speciation rate variation, and what this allows us to do is design studies that maximize our statistical power. We can choose taxa to study that essentially maximize the amount of phylogenetically independent variation in speciation rate that we want to explain. So this is in some ways a really good potential system that we could do a better job of exploiting. And I think that with effort, we can measure many potential components of reproductive isolation for individual species. And I think that when I think about the other fields like physics and the geosciences, for example, they have a lot of the recent insights in these fields have come from using these sorts of big science approaches, such as work on gravitational waves, or in fundamental particle physics, or the search for early life on Mars, for example, with Perseverance. And so I do think, though, that big speciation science would be expensive, probably much less so than the Large Hadron Collider, but it would more fundamentally perhaps require transforming our culture of individualism within evolutionary biology to one that looks a little bit more like some other fields at the moment. But perhaps this is our best chance for serious progress on the mystery of mysteries. So thank you for your attention.
Thanks, Dan. That was terrific. If anyone has questions, please put them in the chat. So we have a question here from Emily. Is there a clear relationship between speciation rate and other micro parameters like recombination rate, de novo mutation rate? I just lost my chat. Genome size and linkage disequilibrium.
Sorry, I lost my camera. There we go.
Yeah, you're back.
Had to switch computers for it, but so let's see. So is there a clear
relationship between speciation rate and other micro parameters like recombination rate, de novo mutation rate, genome size, and linkage disequilibrium?
I would answer no, although I would also say that we have not done a very good job of looking at it yet. So that is an area where we could do a much better job, but I would say that so far, I mean, as Hélène pointed out, there is evidence for connections potentially with genetic diversity. So these are cases where, and perhaps there are yet other genomic mechanisms that underlie that phenomenon, which may be more population level aggregate phenomena that have to do with the nature of range expansions or non-equilibrium demography or fundamental features of the biology of organisms that shape their actual standing patterns of genetic variation. I think we don't have a clear answer to that yet. So I would say, though, that from a broad sense, so if you look at the evidence for some of the amount of variation in things like, say genetic variation across the tree of life, and you look, there's a really nice review from Leffler et al. From 2012, looking at just the magnitude of genetic variation across different groups of organisms. And it's remarkable how within some of these, and I expect that some of these other processes that have to do with recombination rate and so on would be similar. There's a lot of variability in some of these things, and I will be surprised if the fact that we have a lot of overlap in some of these parameters like genetic variation and the across very different groups of organisms. So like arthropods overlap very substantially the amount of genetic variation, for example, that you see with invertebrates, even though you might see some differences. So it might be there might be more variability there and the genomic mechanisms than there is with the speciation rate. So, but I guess best I can say at this point is that a lot of these are open questions.
Great, thanks. And then a question from Katerin. What about specialist and generalist species? Can we predict speciation rates based on this? A hypothesis about this proposes that specialist species tend to speciate more than generalist ones.
I mean, that again is a really good question. So I would say that this is a case where there is a fair amount of theory on that as to one might predict some of these types of things. But I would say that the evidence has been so mixed, and lots of folks have looked at this in different contexts, from plant feeding insects to other types of lineages that are specialists. And I would be surprised if this effect pans out more generally. And I think that even when we look at the scale at which there is that speciation rate variation manifests itself within clades that we are relatively well studied, like birds and squamate reptiles, for example. We see that there are clades of organisms that have consistently fast or not fast speciation rates, and you can just look at the amount of ecological breadth and how it varies across those taxa. That doesn't mean that we haven't yet identified some particular sort of context dependence to that would sort of perhaps give it more predictive utility, but I would say that thus far we haven't identified a general signal of that. And of course, You can even make, this is one of those cases also where the predictions themselves aren't clear, because sometimes we find that some of the predictions that we would make about how something relating to specialization might facilitate speciation might also, for instance, increase the potential for extinction of an incipient population. So I guess the point being that sometimes the predictions aren't necessarily very clear as well.
Thank you. So let's take one more Dan specific question and then we'll move into the broader discussion. So Silu is asking if microevolutionary and I think this is actually really interesting broadly. If microevolutionary processes are unreliable for predicting speciation, is macroevolution of speciation rate really reliable? Penel and Luca showed that speciation rate is unreliable neither.
So that is a good question. And I think that there is a lot more to be said about that. And I expect it is likely the case that both Hélène and I have a lot to say about that specific topic. I would argue that I think it is yet to be demonstrated that speciation rates near the tips of phylogenetic trees are especially unreliable. I mean, I have had concerns about estimating evolutionary rates, at long time scales for some time, and I think that the evidence that might be problematic goes back a bit deeper even than Luca and Penel, but I think it's a good point and is a fair point. I would say that we are still in maybe the early to medium stages of grappling with this idea of where our inferences break down. I mean, so I don't know. I think that's maybe a point we can take up during the expanded discussion. But I think that, I guess from my perspective, I think there are some things that we can know better than other things. But I think there are. I wouldn't do a blanket dismissal, I guess, of everything based on that result, for example. That's how I would leave that for now.
Yeah, I guess. Sorry, Liz. I was just going to say, we're kind of in the discussion session part of the thing. So maybe, Helen, do you have like a perspective on that question, the kind of Luca and Penel issue?
I was happy that Dan was the one to answer. And I also wished that we would talk about the macro question and not this question that has taken a lot of room in the discussion. And I agree with Dan that there's still a lot to be said, and it's not time yet to dismiss all our the trust we can have in some of the estimates we get. Of course, there's a lot of limitations. It's estimates from models that are fitted with data with some assumptions. And so we always have to keep in mind that these are estimates under some assumptions of models that we fit to the data. So they are not to be taken at face value, but it's yet to be proven that they are not reliable at all, or that we cannot have interesting and accurate inference, for example, of correlations between macroevolutionary estimates of reproductive isolation and genetic differentiation and so on, and speciation rates. Nothing has been said, for example, about heterogeneous models so far,
Yeah.
So whether we can at least have a good estimate of, which lineages speciate fast and which others do not. And I think we'll still have a lot of power of inference from phylogenetic information from this data.
I'd make one follow-up on that, which is that there are also some other areas, even if I am somewhat optimistic about some areas where some of our inferences might be more robust, such as towards the tips of the tree. Note that also is what I would call the sort of danger zone for where the biggest problems would come up with what our definitions of species are, and things that we might not know about the issues that have to do with taxonomic uncertainty and even how we conceptualize species. That is the area that brings into this whole other set of potential problems. So I guess it's not necessarily the case that just by looking at one particular area we make certain problems go away.
Go for it, Liz.
So I think that does segue into a question that someone had asked after Helen's talk that we didn't get to, which is that how are you defining species in these models?
So in the, I believe this relates to empirical analysis on mammals. So in this case, we are lucky, right, to have, it's mammals, and here we really rely on expert taxonomists on mammals, the most updated taxonomy of mammals, which might change, which is changing, but that's the definition of species that we use here is from those mammalian experts. Based on taxonomic database. Now, I think there was a question, for example, there's groups where it would be much more complicated, of course, to come up with a species definition. In this case, what we can do is use different definitions. For example, when we work with, we have some works on fungi, for example, where we use different ways to delimit species, which all have their own weakness and strength, but we can see how robust the types of correlations we see or patterns we see, how robust they are to these different species definitions. So that's the best we can do, I guess. In the mammal study, it's not as big as a problem.
I mean, I would